Our next speaker is Ilana Fertig. Ilana is an associate professor of oncology at Johns Hopkins University. And since 2021, she is also the director of the Division and Research Program in Quantitative Sciences, the co-director, Convergence Institute, and associate director of Quantitative Science at the Sydney Kimmel Comprehensive Cancer Center, also at the Johns Hopkins University. She has a PhD in applied mathematics from the University of Maryland, where she also studied her master's in mathematics. And today she will talk about transfer learning for transcriptional regulation. So the floor is yours. You can share your screen.
Wonderful. Thank you so much for having me here today. It's an honor to be here. And I'll be talking today about some of the work that we've been doing for transfer learning in the context of transcriptional regulation. Though, of course, a lot of the transfer learning is generally applicable to other questions in multiomics in addition to epigenetic regulation. So because of that, I'm going to start today by really talking about single-cell RNA-seq as our baseline, and then talk about how we expand this concepts of sort of pattern detection and transfer learning from single-cell RNA-seq into the space of ATAC-seq. So why starting with RNA-seq? And I think this also applies to single-cell ATAC-seq, but exactly like the last speaker and Maria were saying, just the sparsity of the data, I think, limits some of the signal currently. And so when you're looking at transcriptional data, I really view that as sort of the hub that you come in, where you're able to get a lens that then you can overlay different sources of regulatory information. And why is that? It's because if you look at the transcriptional signatures, and Aviv Regev did a great job of summarizing this in this review, the transcriptional signatures really give you different axes of cellular identity, where you're able to get out what are the different cell types, what are the phenotypes, what's the temporal progression, what's the state, and what's the spatial position. All of these are hidden in single-cell RNA sequencing data within the transcriptional profile. And so the trick becomes, how is it that you can learn from the data what these are in order to figure out what the dominant features are, and then overlay other types of data modalities, such as ATAC-seq, to figure out what's regulating it once you know what the underlying processes in the data are. And the trick with that is that if you get a lot of data sets, the reason that we're doing this single-cell RNA sequencing is that, frankly, we don't know what's there, right? So we can have these hypotheses about the axes that we want to learn, but in reality, we're trying to learn that from the data. And so a priori, we don't know what's going in. And it's very important to have unsupervised learning algorithms that enable us to get that. But in order, before we can go to the place where we don't know something, we need to make sure that our algorithms are actually reflecting biology to have a baseline and ground truth to benchmark. And so in collaboration with Loyal Goff and Seth Blackshaw at Johns Hopkins, one of the things that they did is they realized that the retina provides a really nice source of sort of a semi-ground truth. Because if you look at retinal development, the order in which cells develop is very well established in time through low-dimensional experiments. The cell types that come out associated with development are very well known, and it gives a model for the larger central nervous system. So it's a really great model system with well-established biology that enables us to benchmark. So what Seth and Loyal did is to collect over 100,000 cells over the time of developing mouse retina, looking across different age points and different cell types. So there were 12 ages, all spanning developmental time, and then performing clustering analysis, able to identify the dominant cell types within the retina in the mouse. And so now we have this big data, we have the UMAP. So the question becomes, how do we start learning these axes of cellular identity? And so if we think about this data where we're getting the transcription, when we have the data at the end of the day from single cell analysis, we really still have fundamentally a matrix of data, right? Where we think about it in these complex embeddings and these complex states, but fundamentally we have a matrix where we have genes along the rows and cells along the columns representing different conditions. And if we're trying to figure out the underlying dimensions of the data, it's pretty well established computationally that matrix factorization provides a very robust set of tools to do this, where when you're applying matrix factorization, what are you doing? You're essentially getting two lower dimensional matrices that summarize the patterns in the data, right? One of which that's associated with low dimensional subclusters in genes, which we can use for gene set discovery, pathway discovery, or biomarker discovery. And then another one that distinguishes the cells that can be used for clustering, subtype or subclone, and time course analysis. And so the trick becomes, which matrix factorization or which factorization approach do I use, right? And UMAP is essentially another dimension reduction, which is very useful for visualization, but I would argue a little bit less so for the underlying biological signal in the data in absence of clustering overlaying it. The challenge with clustering, of course, is that you're assigning every cell and every gene to one group or another. But of course, if you go back to the UMAP, right, there's some notion that you have a temporal process that's associated commonly across different cell types. So there can be a great desire in biological systems to try to find not patterns that are unique to one cell type or another, but patterns that also overlap between. And in particular, if we want to figure out these different axes, that's going to occur a lot, where we have different types of data that are occurring, where we have different signals and different genes that are being reused. And fundamentally, non-negative matrix factorization lets you do that. So if I pretend, instead of being in this whole high dimensional world of, 20,000 genes by 150, 200 million cells, whatever we're getting to now, to my cartoon world where I have only 4 genes and we're trying to figure out what's going on in the signal, I've plotted in this cartoon on the y-axis the expression of each gene, and on the x-axis you can imagine it's each cell. And in this simple cartoon with my 4-gene world, I can very clearly see my axes of cellular identity. Well, there's only 2, right? There's one thing going up and down, one pattern going up and down in the first set of cells, and one pattern going up and down in the second set of cells. And each gene is either using one or the other to different degrees. And so non-negative matrix factorization lets us represent these axes with addition of genes associated with each of them, so that we can specifically model overlapping processes that co-occur in multiple phenotypes. And then for non-negative matrix factorization, all you're doing is scaling it up to higher dimensions. So instead of my cartoon, now I go to my whole single cell data. So we've developed a particular Bayesian sparse non-negative matrix factorization. And the advantage of this model is that it fundamentally models the sparsity of single cell data, because we're using these gamma priors in the data for each matrix element. So what does the gamma prior do? If we think about the gamma distribution as a sum of exponentials, what that's going to do is, if we have a low value of the hyperparameter here, it's going to tend to look like an exponential. So it's going to constrain it to a very sparse signal. And as this hyperparameter gets larger, it's going to actually constrain the data away from sparsity. It's going to have more of a normal distribution that constrains it away. So the advantage of this particular model is that it learns the sparsity of the data. And if a gene is very sparse, it's going to tend to model that and have the sparsity constraint. Whereas if a gene is more highly expressed, it's going to model that. And this becomes particularly important, as we were talking about in the last talk, with the sparsity of current single cell ATAC technologies as well, that you're really able to model with this one matrix factorization approach, the sparsity of different levels of the data. So now going back to this case of retinal development, if we apply this matrix factorization approach to the developing mouse retina data, what we observe is that we're able to distinguish with one algorithm patterns that are associated with cell types like rods, but also patterns that are associated with transitions in the cell cycle that are associated with the early developmental processes with stem cell divisions in this population. And indeed, we observe around 80 patterns in the data, several of which are associated with the transitions, like I'm showing here for the neurogenic cell types and cell cycle, as I was showing before, and also several patterns that are unique to specific cell types. So with one approach, by using matrix factorization, you're able to concurrently learn cell types as long as discrete cell state transitions. So the next question is always, well, how do I know any of this is real? It's helpful in the case of the retina, again, where I have this ground truth, but how do you know that this is actually reflecting any sort of biology? And so in order to do that, we developed a second transfer learning approach, with the idea being that if we think about these transcriptional signatures that are learned from matrix factorization as the lenses through which you view the data set, now if you have a new data set coming in and you project it onto those transcriptional signatures, you can ask, where does it occur in a new data set? And we think about this as a form of in silico validation of the patterns that we're getting, because if we are able to apply this to a data set with a similar biology, then ideally the patterns that are associated with technical artifacts or batch won't project because they have nothing to do with the biology, whereas the ones that are associated with biology will. And so as an example of that, we applied transfer learning of the rod-specific pattern that we learned in our development data set to a data set of the mature murine retina, the adult mouse retina. This was a different mouse model, and it was a different single cell technology. So we used 10X, they used SmartSeed. And what we observe is that when we apply transfer learning, the rod pattern that we associated with mature rod cells learned directly from the single cell data perfectly maps to the rod cells in the mature mouse retina, showing that we are indeed able to preserve cell types. And we went on to show several other state transitions as well as cross-species analysis, mouse to human. But the focus here and of this meeting is largely epigenetic regulation. So what does any of this transfer learning have to do going between mouse and human, or sorry, going between different data sets with that epigenetic data? And so where we can use ProjectR is anywhere that our genes or our features are very closely related. So we can learn the patterns on a source data set and project them into a new one. And so the obvious cases, like I showed you, are where you're taking genes from one and samples from one context, and then you're going into another. But we can also apply this in the case where we have matched single cell RNA-seq and single cell ATAC from similar conditions, right? It doesn't need to be exactly the same cell, but similar conditions, because the features are related in terms of the regulatory relationships. And so as an example of that, in this case of the developing mouse retina, we had matched bulk ATAC-seq even of the murine retinal progenitors over the developmental time. And what we observed in the original one is that if we just look at the average log2 binarized expression or log2 binarized counts of the peaks, we observe these different peaks that are sort of co-occurring in levels of regulation. And when we project onto different signatures and associated with the patterns, we observe some that have nothing to do with epigenetic regulation that are relatively flat and noisy, right? So we're not seeing the epigenetic regulation. But others, for example, were able to distinguish over developmental time and line up replicates that are associated with regulation of distinct patterns by projecting the patterns from single cell RNA onto the single cell ATAC data. So that gives us one means of seeing which of the patterns that we're observing in the data are likely due to epigenetic regulation. And by going back to the features of that, you can link that to specific chromatin elements. So we wanted to go a little bit deeper and say, well, what if our focus is really the transcriptional regulation and not just the sort of gene expression changes that we're modeling with single cell RNA-seq data? So what we can do is, again, if we're using single cell ATAC data, we're getting out another matrix of data, essentially, where it can be either features that are learned across peaks or DNA motifs or any sort of other summary that you want to get of your epigenetic data. And so just as we were applying matrix factorization in the context of gene expression data, now we can apply it directly to single cell ATAC data. And so now what we're going to do is when we get out these patterns in these gene weights, we want to figure out what the regulatory elements are, right? So we don't just want to look at these whole gene signatures like we were in transcription. We really want to figure out what are the regulatory elements when we learn these patterns. So what Genevieve Stein O'Brien from my group realized is that a lot of times when we're applying matrix factorization and developing markers of genes, what people tend to do is focus on which genes are most highly expressed and in a pattern. And what's tricky about that is if I'm trying to sort of map these unique regulatory processes that add up to the data, if I look at the most highest weight gene, in this, even in this simple cartoon, you can see it would be this one for the second pattern. But this one really is using both patterns. And so what's more unique to it is actually this first gene, because it's only being used in the second pattern. So what Genevieve did is to develop a new pattern marker statistic that would basically highlight the first two genes that are uniquely associated with one pattern or another, and then use that to define a pattern defining feature. What Ross and Urbe went on to further do is, in the context of epigenetic regulation, realize if we're applying matrix factorization to learn what's going on through epigenetic regulation, we should identify these pattern defining peaks through the pattern markers. And then you can figure out which genes are mapping to the peaks by looking at how close they are to a transcription start site and perform gene set analysis to learn their function. Or you can match them, get a set of genes that are associated with a pattern, and then perform motif analysis to figure out enrichment of transcription factor binding. If, on the other hand, you're applying matrix factorization directly to motifs, then you can use these pattern defining, your pattern markers instead will be applied to motifs, and you can use that to find enrichment of transcription factor binding sites. So what Rawson did is again, we wanted to have this sort of ground truth. So Rawson took a public domain data set from Shep et al. That had done single cell ATAC sequencing profiling of different cell lines. So we knew what the underlying condition was. And he applied COGAPs in this context to this data set, and then performed either the motif enrichment or the gene set analysis to look at function. What he observed is that when he applied the matrix factorization approach to features that were associated with peaks instead of motifs, he was able to derive very clean signatures that isolated each of the different cell groups that were in this sample. And one pattern, for example, that was associated with the erythroleukemia cells, then he was able to link that to processes that would be expected, like heme metabolism, for example, through the gene set analysis of the pattern markers. On the other hand, the other pattern associated with monocytes was then associated with a large number of immune pathways, again, making sense in the context of this. What was interesting and unexpected to us is because there's been a lot of call for performing single cell ATAC analysis in the context of motif features instead of peaks, is that when he applied the same approach to motif level data and tried to obtain patterns, they really no longer distinguish the cell types anymore, which we were not expecting. But what did occur in this case is that he observed one pattern that was linked between different erythrocyte populations that spanned the cancer and normal erythrocytes. So he was able to define a feature that better segregated overlapping groups of cell types that wasn't learned through the pattern detection on peaks alone. And so what we think is going on is that if you apply your data, your matrix factorization to peaks versus motifs, you're fundamentally learning distinct features that give you different bits of information about the data, with motifs probably providing larger summaries of what's shared between different processes, but peaks giving you more of an ability to delineate. And so again, our question is, well, how does this relate to transcriptional regulation, right? Because ideally, we also want to look at this in the context, or sorry, how do we sort of look at this? So
Ilana, you have a couple of minutes left.
Okay, perfect. I'm getting to the end.
Perfect.
So what Rawson did is further apply transfer learning to map ATAC-seq across data sets. And so taking this between this, he was able to relate the monocytes that he learned in the one data set to monocytes from another SCA TAC data set. And then again, apply this erythroleukemia and see that it separated erythrocytes in another data set through projection. And this was taking ATAC-seq to ATAC-seq. He went on in another data set with matched ATAC and RNA to show that the transfer learning does enable you to go in the other direction from ATAC to RNA, just as in our previous study, we were able to go from RNA to ATAC. So in summary, matrix factorization are really robust tools. Single cell COGAPS discovers meaningful patterns, both from single cell ATAC and single cell RNA data. And the concept of transfer learning really enables to explore data sets for biological reuse in the context of epigenetic regulation. So with that, I'd like to thank the members of my lab. Sorry, I don't have a picture of Rawson up here, but everybody who contributed to the work and our funders, and I'd be happy to take any questions.
Thank you very much for a very, very nice talk. I think we have one question on the chat. I will read it. So it's from Tule again. What are the underlying assumptions to make sure that the cell labels can be successfully transferred to the new data set? How does the method handle the batch impact and cellular composition difference between the original data set and the new one?
So that's a great point. So I will say we don't, our attempt is not to transfer cell labels. It really is to transfer the pattern. And so one thing that needs to be done is there does need to be some independent notion of labeling, right? You need to do clustering, you need to label them somehow. But once you learn the features, then those are what you're trying to project. And so the idea would be that ideally you would have cell labels that you could go back and forth. What we found is that the correlation structure that we learn does tend to be very robust between data sets in spite of batch, in spite of cellular composition. And so if you're learning it on a large enough training data set, what we found is that the features that are associated with biology do transfer. Whereas in our data, I obviously didn't highlight the features that were associated with read depth and batch, but they are learned directly in the data, and those don't transfer to a new context typically.
Yeah, thank you very much. So do you have any theoretical explanation for that? I mean, observation. I mean, the features are somehow quite robustly, learned.
I mean, we think basically because if you imagine you have a transcriptional program, right? That's going to give you a certain correlation of the genes where you're going to see that they work, where you see they're all going to be correlated together and having a similar profile. If it's batch, it's not going to have that same structure in a new data set, right? That's going to break the structure in a new data set. And so we think that's why the correlation structure, we're basically mapping the preservation of the correlation structure.
Yeah, I see. Yeah, thank you very much. Great talk. Thank you.
Thank you. We have another question from Ken Wang. You can unmute yourself.
Can you hear me?
Yes.
Yeah.
Yeah, I guess a very nice talk first. And I have one question, maybe I missed it, because you have the grand choose data to validate whether each dimension dimensionality have the biological sense or biological meaningful some sort of things. So I was curious about if you ever develop such kind of statistical metric that assess each one of the dimensionality when you try to apply your methods on the tissues that you don't know about the grand truth biology. So you actually could assess like which dimensionality that you should look at first and which dimensionality you should look at the last, like something like that. Because you know the PC actually you could using the eigenvalue to determine which dimensionality is most significant.
So I'll say, so there's two sort of bits in there. How do you know which features to use, and how do you know like the dimensionality, right? So for dimensionality, we specifically use NMF because we think that it's capturing features that reflect different biological processes. And so the trick with PCA, right, if you're using PCA, there's a notion of how many dimensions you should look at because you get this elbow plot.
Yeah.
But it doesn't actually give you the biological features because the features that are highest variance, right, will be fundamentally mixed together, right? If you think about it like cell cycle.
Yeah.
And so that makes it a little bit tricky because we don't have an equivalent of that. And one of the things that we're moving more and more towards is a multi-resolution model where we actually look at multiple dimensions. And the reason we do that is, let's just take the example of T cells, right? You can imagine a low dimension, they would be leukocytes. Then you go to T cells, then you go to CD4, CD8. There's actually a hierarchy of number of dimensions in the data that's embedded in the biology. And so we actually don't think there's one right dimensionality. We actually think the right thing is across them. The concept, I think, with transfer learning, what it gives you is if you have a really robust set of features in one data set, one of the things that we developed in our paper is a classifier of basically how well patterns transfer from one data set to another. And other people are using this in reference atlases now. And so what we think is if you have a database where it's really well annotated, and then you can use the transfer learning and the features learned and how well they project on another data set to highlight which cells and which features you should be focusing on in a new data set where you don't have the ground truth.
Okay, that's great. Okay.
Thank you. Jörn, go for it.
This is actually a point with non-negative matrix factorization that for the interpretation you need some references, and that makes everything so difficult. Particularly if you have, as you said, you have an intermingling of different features that give you clear classification. And I mean, we are at this point with the DNA methylation, for example, deconvolution. But yeah, can you comment a little bit more on that? So how do you want to approach that in the future?
Yeah, so that is really where we view the power of transfer learning, right? Is that you can take it from something where you know a ground truth and ideally use that to help you annotate. But I agree with you, the visualization and interpretation of non-negative matrix factorization is really, really tricky. And so that's, I think we need more software tools, and this is something that we're sort of starting to work on some workflows in my lab, that there's a lot of tools for visualization of genes. There's a lot of tools for visualization on UMaps of features and things like that. There aren't a lot of tools for visualization of patterns, visualization of features. And I think as it's becoming clear, be it non-negative matrix factorization or whatever latent space method you like, it's very clear these unsupervised learning latent space methods are here as a central toolbox for single cell analysis. And we really need statistical measures that are overlaid, that are generic across latent space methods that let us interpret and annotate these features, be it either through the gene correlation signatures, through transfer learning like I showed today, or post hoc statistics of like differential expression between groups, between phenotypes, and ways of viewing that.
Good, thank you.
A lot of work.
Yeah, but this sounds really super interesting and necessary indeed. So very nice. I have a couple of questions, but for the sake of time, I think we need to move on. Thank you very much, Ilana, for a great talk.
Thank you for having me.